it's not as important in this class as the antibiotics we just talked about, or you just answered some questions for. And then for the parasites, you have anti-parasitic chemotherapy. So malaria, for example, fits into those. Quinine, chloroquine. There are some. So those are mainly protozoans that you're giving drugs against. Some of those, see, like tetracycline, you've seen before, sulfonamides. So those are some that also are used for bacterial infections. And then when you go further down to the worms, the drugs that you use there sometimes have specific actions, like this one. It destroys the scolex, so it's a part of the worm's morphology or anatomy, or it paralyzes the muscle. So there's different compounds for these three groups, but you can maybe look at those and have an idea of maybe one or two compounds for these. Anti-malaria, malaria is a pretty big one, just because malaria is still a disease that's been a problem, and there's a lot of anti-malaria drug research, and they're trying to get that under control, which it's still one of the main diseases worldwide. So those are more important, but you can see that those have all the clean endings, so you can memorize, or you can recognize those. But that just should wrap up the different chemotherapeutic drugs that are available for these specific microorganisms. And so what's important is really the main idea, and I wouldn't go any further with what I've asked you just now in the quiz. Like, those are the main groups for the bacterial infections, where you know what they target, and maybe a couple examples for those, and then just the basics for the eukaryotes. I don't want to emphasize those too much, because it seems like to me, when, at least you get some kind of idea how bacterial infections are fought with drugs, but with these, if you look at the variety and the amount of protozoans there are, and then you'll memorize one way and one drug, it seems like it's useless to me. But they exist, and you should have heard that that there's drugs for those as well.
Is that quinine really discussed in the tonic water?
Sorry?
Is quinine really discussed in tonic water? Is tonic water really has the same quinine?
Tonic water?
Yeah.
I don't know. I would not. No, it must be. I don't know what the compound in there is, but that shouldn't be the same.
Okay.
So the last group we're going to go to, so we have looked at bacteria, prokaryotes, and then we always had bacteria, and then we had the eukaryotes, basically, and then there was one which, obviously algae we leave out, but there's the worms. I'll make it short, the protozoans, and then what else did we do? Fungi. And then there's one separate class, and that's even, or one class of or group of microorganism viruses, which are so different from those, because there's no cellular structure, that it gets even more complicated. And you can imagine, because you're trying to fight something that's actually reproducing in a cell or reproducing in the host. if you try to block or if you try to harm that virus, and you apply something that works against, say, the replication, or that uses a metabolic system, then you're basically harming the host automatically with it. So the main reason, or the main problem here is just like with the eukaryotes, where you have already a problem of similarities in the cells of the host and the microbe. Here you have the problem that the microbe is actually using the host cell as a place for replication and synthesis. You already know about how that works, and so the main modes of actions are to block the penetration into the host cell. So before it actually gets into the host cell, but when it's entering, then blocking the replication, transcription, translation. So basically, once the virus is in, and the genetic information is ready to get read by the host cell, you block that, and that works a little better because, say, you have an RNA virus. There's different enzymes involved in the replication, transcription, and translation of viral genetic material than the host one, and I'll show you one. So there's the difference where you can try to block, and then also preventing the maturation of viral particles. So if it's produced, maybe the exit of the virus, make that impossible, or block enzymes that help, for example, the cutting of the genetic material that has been produced. So when the host cell produces these long strings of genetic material, or of protein, essentially, after it's gone through all this, it has to be, remember, it has to be packaged and everything else, and so you can interfere with that. And why that's important is a lot of times, or why there's a lot more knowledge is mainly for HIV, because it's a global epidemic, and so because it's such a dominant disease in our time, there's a lot of research going into how do we block a virus like that. So that initiated a lot of research. So if you go back, if you start off with penetration into the host cell, influenza is a good example for that. And you have these two compounds, and what it does, it prevents, and focus on these two. And I don't know if you can see it. It's in your book, so if you have your book here, you can open it. Basically, these compounds are blocking the entrance, the fusion of the virus with the membrane. And remember that the envelope viruses are able to fuse because their envelope is full of lipids. So you have the ability to fuse, and if you have basically proteins blocking that the ability of the virus to fuse with the membrane of the host, then you're blocking the entrance. And that's mainly shown here. And another one is it could also block the exit, because it does the same kind of thing. And a lot of times what the virus needs is a certain neuraminidase, for example here, a certain enzyme to get into the cell or out of the cell. And if you're able to block that, the virus is not going to be able to get into the cell. So that's one way to do it. And influenza is usually the medications or the drugs you would get for influenza are likely to try to block the entrance of the virus into the cells. And then if you have some that are actually blocking, as I said before, the replication. And in this case, it's an anti-herpes drug. And what it is if so, say and focus on this one here, the top one. So the virus in this case gets in, and you have the virus DNA, and it enters the nucleus where the DNA is replicated, and then it can actually block this DNA polymerase. So it blocks this enzyme. And remember, yesterday we had an RNA polymerase in that construct, in that plasmid, that was initiating the reading of these enzymes to convert the sugar or ferment the sugar in this plasmid. So that's the same structure, it's just a DNA polymerase, and this drug is basically occupying active sites there, and so the DNA polymerase is unable to replicate DNA, and there's these different compounds that all do the same thing, or that are all targeting the DNA polymerase. Another interesting thing is that sometimes, and it's almost outsmarting the virus that way, sometimes these compounds are activated by an enzyme that's actually produced by the virus. So you can then say it makes it more specific, because the virus is, when the virus enters, only if the virus is there and produces their own enzymes, you get an activation of these, and then it blocks where the polymerase where the virus has already invaded the cell. And so there's different oral and topical treatments. So on the skin and oral treatments for herpes, chickenpox, shingles. That's kind of like what that group of drugs does. I'll just go back there. So you block the penetration into the host cell or the exit. Sometimes happens both ways. You can block the replication, transcription, or translation, and those examples would be influenza and herpes drugs. And then for HIV, because it's such a, there's basically what researchers trying to do is to find many different ways. It's not as easy as finding a drug that works on one specific site and you're done with it. So they're trying to do, over the years they found various drugs that work in different specific, will have the specific action, modes of action and specific sites. And some of them are just like what we've seen for influenza, for example. So they inhibit the virus entry. Fusion, Marowak are two of those. And then some of them, for example, inhibit the assembly and the release, and those are protease inhibitors, Saquinavir, for example. And so what they do is just like in the same, or just similar to what we've seen. You have a protease that basically then if you have the protein that the virus produces, it would cut that protein into the right pieces, and it would be assembled, and the virus can then leave the cell. And because these proteases are inhibited, the virus is never going to be a functioning particle, not organism, but yeah. So the virus will not be functioning because of that. And then there's two other things that are specific for, or that are used as a target for AIDS or for HIV, and that would be either, and you can see that in the book too, it interferes with the viral DNA synthesis, and it's either, again, this transcriptase is either directly inhibited, it's done by using nucleotide analogs. Retrovir is one of those, so it it would put nucleotides in the production that are not virus specific. So it would use analogs or similar, almost like a competitive inhibition. So it would target this transcriptase by using analogs with this compound, or by inhibiting some other non-nucleoside inhibitor. So either an analog or something that blocks this transcriptase. So those are the three, and it's just important to know a little bit about that, because it is a pretty important disease still, and so there are different ways, and I don't know if anybody knows what the state is right now in terms of what you would receive. I heard, I think yesterday on the radio that there was something really interesting today, but I couldn't. I don't think I can reproduce what I heard. So, but I think for HIV, they wanted to. So they basically do a cell count, usually because it attacks your immune cells, and they count a certain cell type. And so what they're trying to do is now lower that. So in I don't know what the numbers exactly are, but for a healthy individual, but it used to be, I think, that the cell count of 500 or under initiated this process of treating you, because you would already see the virus affecting your blood cells enough, so this lower than normal 500 count would initiate the process. And in South Africa, I think must have the highest percentage of HIV infected people, I think, worldwide. And so now they're trying to lower it to 350 to say we're going to start this medication earlier. No, that has to be the other way around, because that would mean 350 would mean this is lower. So it should be. It used to be. That's when you try to reproduce what you heard on the radio. I get my numbers right. But anyway, so they're trying to set the limits up a little bit. So if say it used to be 350, so all the way down to 250, you would start the medication. Now they say, well, let's start earlier, so we give those people a chance to get treatment beforehand. What the problem is that they say, even for the people that were so far, the people that are trying to get into the system, it's so expensive, it costs so much time, it's such a big operation that so many times people come, they travel for days to just go to the hospital, and they say, we don't have any medication, come back in a week. And so they say, number one is, first of all, you have to make sure the medication is available to all the people that need it, that are actually already in this bracket. And the other thing is, if they think, one researcher was concerned about doing that because he said what's known about these medications is you get like a cocktail, and it's toxic, and it has to do with the toxicity comes, a lot of the toxicity comes from the duration you take it. So it's on a short-term basis, it's not so toxic, but over long periods of time it is. And so if you start people early, you might expose them to these medications a lot longer, and so they might show more of the problems. And I don't know what they exactly are, but they might be affected more by the side effects of the medication rather than their actual disease. So those are just some of the controversies, but it's still there's, I think at the end of this today's lecture, there's a couple slides about it's in the section of epidemiology, so monitoring where the disease happened, what the disease are in this country, for example, and there's some maps of how many cases you find, say, in California, Florida. I just remember Texas, California, and Florida were dark purple, which meant they had more cases than Montana for example. What else is? But then I always think, like, is there anybody in Montana anyway? Who can get sick there? Yeah, I spent a little bit of time up there for research, and it was, I think it was still is it under 1 million? I mean, it's like, you can fit 2 Germanys into Montana, and there's like 800,000 people, which is, you know, probably a little village in Germany, 800,000, yeah. So it's empty, but they definitely don't have a huge demand for these drugs up there. So, what do you think, from what you've heard? You get that early? What do you think? Where do viruses usually get stopped? So we see we see that in terms of using proteins or drugs that actually attach. So you see blocking the virus receptors. The maturation, we talked about that. Stopping virus synthesis, we talked about that. So A is the right answer. So now back to what we talked about a little bit last time, the acquisition of drug resistance, and that's another huge problem. And what's interesting is I don't even listen to the radio usually. It's just because I have to drive into this beautiful area over the hill, go from depressed to happy, go from a sun depression to a happy fog. We talked about this when we came in, and I hear stuff about disease all the time, and I think, you know, when you're not really taking, I mean, you're probably already a little bit more, you're probably a little bit more interested in initially, but it really is amazing how often there's talk about different drugs, about disease, and it makes sense, you know, it's really something that if you're healthy and you're not in the field, you probably never think about it. And if you start, you know, either taking a class that deals with it, or you want to get into that profession, you start hearing about it, and you think every day there's something on it. They're not, you know, they're talking about, today they were talking about Monster Barbies or something. I mean, they talk about everything, you know. It's not that they're do you hear any about that? No? There's apparently some, you guys don't have girls between 6 and 12? Apparently the new Barbie is Goth Barbie, and they're unbelievably successful. So that's the stuff you hear on that radio station too, which means how come disease are always up there, but it's because we have a lot of issues. Monster High is what it's called, yeah. You know anything about that, huh?
No. I got a boy.
Well, I use 5 too. Apparently the group that buys it is between 6 and 12. But they're out racing, out competing Barbie big time. Just that's also on that radio station. But there's a lot of talk about resistance, about disease, and epidemiology was today about how countries, for example, have lists, and they have lists of disease that they're really focusing on, and it's a worldwide operation where you try to monitor what's going on, where you have outbreaks. So it seems like it's a big effort, and what I was trying to say is that the general public, if you're not really involved with it, might not ever hear much about it, or you might not realize how much is going on. So resistance is really a very important one that a lot of people are very concerned about. You could probably have somebody come in today and talk about how close we are to getting completely wiped out because of the resistances. And I've heard that before, but at the same time, you can always get somebody who gives you a really bad scenario for any kind of topic. But it's a big deal, and what it really is you have a microorganism that's able to tolerate an amount of drug that ordinarily would be toxic. So that's the main definition for it, and it's due to the genetic versatility and variation. So you have variation in the genetic code, but you have also it's part of the fast reproduction makes bacteria or enables bacteria to get resistant quick. It can be intrinsic as well as acquired, so it could be you know, if you have a bacteria like a bacillus, for example, producing an antibiotic, obviously it needs to be, or a toxic compound for other organisms, it needs to be able to survive itself. So there is some intrinsic resistance to that specific antibiotic. Otherwise, it would be a suicidal microorganism. And then the adaptive one is is of a bigger concern, because those are the ones that we've already found drugs, and now we're running out of, or we're realizing that they get resistant to that drug, and we have to find new ones. So yesterday I showed you this slide, and what we did is we got this plasmid that was actually free because it was given to us. It didn't come from a different organism, and we through transformation, we got this plasmid, possibly got this plasmid into our cell. I was looking over to the 37 degrees, trying to see if it's glowing. And there's two other ways. There's transformation and transduction. So there's different ways. Transduction is interesting because it means this genetic material, resistance, is actually transferred by a virus. So the transfer is done by viral delivery, and then the other one is transformation, is conjugation, and we can talk about those, or we will talk about those more in the genetics, the bacterial genetics section towards the end of the class. I mean the end of the whole class. Probably is one of the last lectures. So you don't have to know exactly how that works, but what it really means is there's a possibility that bacteria pick up resistance by free DNA or through resistance in a plasmid, or resistance that is transferred through a virus. And what's really interesting is that a lot of times these biofilms, and you hear about biofilms more and more. I mean, biofilms seem to be really, really important. But there's completely unrelated bacteria living in these biofilms. Remember, like say you have a heart valve, and it creates this film of microorganisms that are all working together and somehow building this complex layer. You can find gene transfer. What they found is there's gene transfer between totally unrelated bacteria. So there are these processes happening, and the problem is if these gene transfers happen, then you have all of a sudden bacteria that are resistant that you wouldn't really expect to be resistant, and so it's a major concern to keep that from happening, or to keep, at least to increase your chances that bacteria don't get resistant, because it affects your fight obviously a lot. And so there's a few mechanisms, and they all should make sense to you from what you know now, from how antibiotics work. So we already talked about penicillinase. So it's an enzyme that blocks, that basically inactivates the penicillin. So you bring penicillin in, this enzyme is able to convert the penicillin, the antibiotic, to an inactive form. And then you can another mechanism of resistance is to decrease the permeability. So the antibiotic, if it's natural or synthetic, it also a lot of times meets a receptor, and if those receptors are altered, the drug can't get into the cell. There's features like drug pumps that are able to constantly pump out the drug from inside of the cell surface. So if those are activated, they're basically, the drug can enter through receptors if they're not blocked, and then they're pumped out. So the drug never gets to the site of where it needs to start its action. There's we have this group, the ribosomal binding site drugs, and if those sites are changed, then your drug can't bind to the ribosomes anymore. That's another possibility of, or some other mechanism they found to that creates drug resistance. And then finally, if you think about the folic acid pathway, and imagine you have these compounds interfering with the enzymatic pathway of producing folic acid, what if that pathway uses a different route? There's maybe an enzyme that produces a different substrate, and then you have enzymes go in that route. So you would have drugs. Blocking one pathway, but another pathway would still be taking over and allow the production of folic acid. So that's the alternate metabolic pathway. And this is a really important one, I think. You have it in your book too, but really what I always try to visualize myself is, if you have microorganisms reproducing in such a fast time. I mean, if you look at your, you basically spread out these SFUs, CFUs, these units over your plate, and then what you see, if you actually see a dot, that's a huge amount of cells. So you can imagine if that happens in 24 hours or 48 hours. In some cases, it goes even faster. If you have such a large population, you can imagine that if a population reproduces so fast, that the generation time is so short, that you're always going to have changes. What happens in the human population takes years and years and years and years, or decades, for any kind of small change, and from generation to generation. And in this case, it can happen in hours. So say you have this generation of non-drug resistant cells, and they're dark here. You might have a couple that are not resistant, so they're not affected by the drug. So those would be the lighter ones. And so you have them in this plate, and then you expose them to the drug. Then it kills all of the non-drug resistant cells, and the remaining ones are able to use, because they have this advantage now, they're able to use all they survive and use all the resources, and they do pretty well. And then whatever is left is usually just resistant cells. So you can imagine, in natural selection, we talked about it, it's usually, because of a mutation or because of a gene you inherited, because of whatever the process was, you basically have in natural selection, you have an advantage, and that advantage helps you to reproduce better, and that's exactly what happens here. They have the advantage of not being affected by your drug, and then they reproduce and have more resources, and they're the ones that survive. So it's a typical, it's natural selection, really quick natural selection. I mean, really quick. It happens all the time this way. I'm saying really quick in comparison to what we usually think. If we think about natural selection and evolution, we usually think millions of years. And this one is natural selection in a very quick time. So usually and we talked about this before too, what we do, and it's not that important. The basics is what would we do to limit that resistance, and one of the things is physicians have the responsibility for making an accurate diagnosis. So it's important, as I said, if you have physicians that give you 100% of the time, or if you have 25% cases are bacterial infections, and they give you antibiotics, and then 75% of the infections are actually viral, you're taking antibiotics that are good for nothing. They might affect your natural flora more than anything, and so it's important that, and I think there's a couple of slides about it. It's important that it's properly diagnosed, and then that the host or the patient actually does take the right amount, etc. We talked about that problem, and then drug research. Obviously, we're trying to find better drugs, less toxic, less. Probably easier to take, things like that. In long term, there's a lot of talk about resistance developing because we apply it to animal feed, for example, because as a prophylactic measure, because they're so dense. If you ever read about how they're farmed, the animals are so dense. You want to make sure you don't have a big die-off because of some infection, so you feed them antibiotics. They get into the water. You sometimes, it can get back into the system. So that's a big problem. Resistance through overuse of antibiotics in animal feed and other areas. And there's a couple more things. So interactions, you have. There's some people who, or a certain percentage, up to maybe 5%, are actually allergic or experience a serious adverse effect to antibiotics. It could lead to damage due to the toxicity of the drug. That's something I was talking about in respect to the HIV treatment. You can have allergic reactions. And one of the main things, and I wanted to ask you that today in the quiz, but then I didn't know if we had really talked about it. I think I mentioned it, the superinfection, possibility of superinfection. And it's kind of like the what we just talked about in terms of natural selection. So if you would have a normal flora, so you have a really, and we talk about this a lot more today, you have your regular microorganism composition. So you have different organisms, and they help you with digestion. They help you in different areas, and then you have possibly one that's resistant to the drug. If you so you have an infection, and it might not even be a bacterial infection. You take antibiotics, and you basically, the infection is up here, but the drug is circulating through your whole system, and it kind of wipes out a lot of the normal flora that keeps all the other ones in check, because there's competition. All of a sudden, these competitors may be gone because of this drug use, and all you have left is the pathogenic ones, and then they have the advantage of spreading, and so they would be able to. All you had is really an infection up here, and you get a super infection, meaning you have a problem in your intestines because of the drug use. So that would be an example of how you can get to a super infection. The pathogenic bacteria kept in check because of the natural flora. You wipe that out with a drug, and then because of the advantage, the pathogen overgrows the rest, and then you have to deal with that. And I think in that case with the person with severe arm pain, I think he by taking, that was one of the complications, by taking all these antibiotics, they also created a super infection And that's actually what almost killed him.
Do you think all that, like, antibacterial hand soap and all that kind of stuff contributes to any of this?
I don't think that's probably a very, if at all, that would be a minor concern, because you're basically treating it on the in a state where the bacteria are far from actually being able to invade, and we talk about that today, how the steps later on, how bacteria move, what infection really means, how they enter the host, how they enter the cell. And so if you have if you in general, you would just basically keep the microbial populations down by those, but I think and they wouldn't, on those surfaces they wouldn't reproduce. So once you at least not in the sense of reproduction where they're harmful. So if you're externally, I don't think you would see you would increase the chances of resistance through antimicrobial soaps, etc. I showed you this, or we already talked about this. So identifying the microorganism is really important. You have to take them before the antimicrobials are given. It makes sense. You go to the doctor if you're already if they just guess what your infection is, what the causative agent or the microorganism is that caused your infection, then your treatment might be off. So the identification of the agent is important before you actually get treatment. And then you could test it, and we've looked at tests. I'll show you a couple more. The susceptibility of that agent to various drugs. Remember the plate? That would be one of them. And then you also have to consider the overall condition, because weak patients, immunosuppressed patients are always there's always more of a potential for building resistance and and also for side effects if you apply antimicrobial drugs. So it's important to know the overall condition of the patient. And as I said, sometimes you have to go the route where you say we're going to have to give something toxic, even though we know it's going to harm the patient also, but it's the only way we can get this infection under control. So these are the tests. I won't say much about this one, and there's just a couple more. What's interesting here is what we haven't really done is, so I showed you some of those, and you measure those, and maybe some of you saw that, but really what I wanted to mention before I leave this is that those measurements then are compared to a standard. So just seeing that, you would say, there's no growth, so there is little resistance, but it's always there are standards out there that determine exactly what because you're applying a certain dose here, etc., etc. So there's standards out there, and that's all you do. You compare the diameter or the radius, depending on which we use, to a standard and say, okay, this is enough to show it's an effective drug or it isn't. And then here it's just so you see there's, for example, 2 antibiotics applied to a plate with, I don't know what they grew on here, but what you can see is in this area where they're both diffusing in, you actually see a bigger lack of growth, and that means that those two might be working synergistically. So those two together have a bigger effect than the individual antibiotic. And then here you can't see that so much, but it's a little bit like the UV picture where they have 2 antibiotics applied, and you see some colonies here, and they seem pretty much the same, but these colonies could indicate that there are already some resistant bacteria colonizing. So you would say you would definitely be careful with this antibiotic and if possible give this, because you already see some resistance for this one. And this is called the E-test, and it's pretty much the same. So we didn't do that here, but these are like little plastic strips, and they have different concentrations of your antibiotic. So it goes from high concentration all the way down to a very low concentration, and you kind of look of, okay, so high concentration would produce this round circle, but because it goes this way, the concentration, there's less here, and you can read then what the minimum inhibitory concentration is. So can you see this? Like, what would you say for this one for IP and TC? Can anybody see that? So yeah, around 1. So here it might be a little bit. Actually, here it might be around 1 too. So it's pretty much the same. So around 1, and I don't know. It's probably micrograms per milliliter, something like that. So this indicates where do you start? Because further down that concentration, the growth is not inhibited. So these strips allow you, these E-tests allow you to do the same thing that Kirby-Bauer disc test does really, and it immediately gives you an idea of what concentration works. And then finally, it's the same kind of thing. You have the smallest concentration of drug. You determine it by doing a dilution series where you basically go from a specific, you have a control that has none of it in, and then you dilute it down from 0.2, 0.4, 0.6, all the way down, and you start seeing where the growth stops, in which of these tubes the growth stops. Or you can do that on a microplate. So these are little vials, and you do that here. And in this case, I think the stop of the growth is indicated by blue, and if it doesn't, it's pink. So they put X's here where it changes, and so that would be your minimum concentration. The smallest concentration that visibly inhibits the growth. So these are just different ways of doing it. And then the last thing about for this one is because it would always be good to be able to be testing this in vivo, meaning in the person, but that's already when you apply it, because a lot of times what you see in these tests might not directly apply to what happens in your body. So if you determine this drug would be good for this person and the concentration is low, this is the drug that we should use. It's not really necessarily the case, because in the body things are different. The physiology plays a role. The overall condition of the patient plays a role. So you might have to use different drugs, a combination, or administer differently. And what they usually look at is the therapeutic index, and it just means what's the dose of a drug that's toxic. So if I know 10 micrograms of this dose is toxic say this 10 micrograms are toxic to are toxic for humans, and then you also know that the minimum concentration to actually inhibit the disease is, say, 0.1, then that would be the therapeutic index. And so if this is really high, that means this is good, because you would have to apply a high dose to actually affect the human body, and then in here you want as low as possible, because the lower this is, the least or the less you have to actually apply or have to administer to get an effect. So the higher this value, so if you say, if you would say, so in the other cases, the toxicity starts very, very at a high concentration, and it actually is already showing effects at this concentration. Then you get a much higher index. So a high index is desirable, and they know those for specific drugs. They know what toxicity is allowed. I mean, you can find toxicity almost to anything. You can find toxicity of pesticides to any animal. They've been testing, saying what is the lethal dose, what's the dose that, or what's the dose that affects. For humans, it's a little bit harder because the studies are harder to get done, but they have an idea usually what is the dose that is toxic and what is the minimal the concentration that has an effect on the microorganism you're fighting.
So you want the lowest toxicity and smallest concentration.
Smallest concentration, yeah, and toxicity, that would mean the level of toxicity that actually has an effect. So if a drug has a high, so the top one, if the concentration of the drug has to be really high to see damage or the toxicity for the human, that's desirable. Because that would mean if you have two drugs and one you would apply the same amount, then you would know if one has a really high level of, you need a really high dose to actually make it toxic, then you would say, okay, this one is safe. You need to apply a really high amount. There's an example in the book if you want to, you can look it up. It's under this index. Now you want to come over here? Or do you want to skip this? Actually, that's a good place to take a short break because we're going to the next chapter. So the MIC, the minimum concentration for to cause inhibition is the smallest concentration.